Hello and welcome to a 4-part mini-series for Memento Malum. In this series we'll cover the history of different pesticides. For our first episode we'll cover insecticides commonly used in agriculture and their history. The first one we'll cover will be synthetic pyrethroids. Synthetic pyrethroids were derived from pyrethrum, an extract of different types of chrysanthemum and chrysanthemum relatives. This botanical spray was first developed to control mosquitoes and houseflies. Between 1910 and 1916, however, the researchers Staudinger and Ruschika discovered the active ingredients of pyrethrum, which was pyrethrin 1 and pyrethrin 2. In the late 1930s, using these principal ingredients were combined with a synergist called piperonal butoxide, which was already used to enhance the performance of natural pyrethrum. Many pyrethroids were developed using pyrethrum 1 and 2 as base chemicals. Now onto organophosphates. This chemical disables cholinesterase, an enzyme that's necessary for central nervous system function to occur. The first known individual investigating organophosphates as insecticides was Gerhard Schrader in Germany. He was doing so during the 1930s, which led the government to try to get him to develop new gases for poison gas warfare. He obliged and created many different types of poison gases such as sarin gas, taboon gas, and soman gas, although organophosphates were not used during the war itself. After the war ended, chemical companies in the United States obtained access to Dr. Schrader's work and patents, and from there synthesized many organophosphate insecticides for use in agriculture, the first being called parathion. The next chemical on our hit list will be dimethoate. This was introduced in 1951 by American Cymid as an insecticide. This insecticide is readily absorbed by plants and degrades quite quickly. It was used to replace the Brussels sprouts insecticide metastiox, produced by Bayer in 1957. And it took off due to its high levels of degradation in comparison with metastiox. The next on our hit list will be phorate, another organophosphate made in 1954 by the company Phymet. It was used as a seed treatment to control sucking insects like aphids, thrips, and leafhoppers. Now on to more recent pyrethroids. As mentioned before, synthetic pyrethroids were first developed in the 1930s, which culminated in the creation of a synthetic pyrethroid that did not break down in the presence of sunlight very quickly. These synthetic derivatives were called alirefrin 1 and 2. They had similar properties to pyrethrins, but were more stable. These effectively replaced organophosphates for most applications in terms of insecticides. And since it did not bioaccumulate like DDT, it became a replacement for that chemical as well. The most recent derivative of synthetic pyrethrum is the chemical telufrin, developed by ICI in 1986, which is used to treat root pests in corn, sugar beet, and several other crops. Now onto the history of neonicotinoids, or neonics. Neonics were developed by different companies in the 1980s as a means of replicating the effects of nicotine on insects in a more efficient and potent way, since there's fewer toxicity effects in mammals in comparison to insects with nicotine derivatives. These are primarily used as seed treatments in cotton and various other crops. This of course led to the death of bees in certain situations, as seen in 2008 Germany. The main source of the reasoning behind neonics being a primary cause of colony collapse disorder is that neonics were not used in Australia and bees are still doing fine there, as well as the beginning of a colony collapse disorder just after using neonic pesticides. Although just like other types of insecticides, there's great variations within the toxicities of these different insecticides within this class. And honeybees are less sensitive to the chemical thiocloprid than other types of neonics. Now onto the pyridine class of insecticides. The first one was developed in the late 1990s as a low-toxicity systemic aphicide. The mode of action is that it causes aphids to be unable to feed within half an hour, resulting in death by starvation. It also works on other sucking insects such as whiteflies. Another insecticide in the same class, called pyroviricine, was developed in 1993 and was used in vegetable crops and ornamentals in greenhouse conditions. Now on to our most recent type of insecticide, the spinosins. It was derived from the fermentation products of the fungal species Stachyspora spinosa found in a soil sample in the Caribbean in 1982. Since then, 20 natural forms of spinosins have been found, and 200 chemicals produced by this group of fungi have been found and later on synthesized. The claim to fame for its insecticide is that it binds to a different receptor than most other insecticides. As of right now, it's mostly used in the UK on vegetable crops, and a few times on apples and pears. On the United States, it's used on a lot of stuff. Pretty much any crop, really. Now on to our last chemical, the diamides. This is derived from the botanical pesticide called Rihanna. This chemical activates calcium receptors, causing a large release of calcium ions, creating deadly muscle contractions. It was used in fruit and vegetable crops until it was revealed that it was very toxic to bees, thus ending its use. Well, that about covers everything. Thank you for watching. Hello and welcome to another episode of Pesticide History. In this episode we'll cover the history of herbicides. We'll start off with the history of non-selective herbicides, which are herbicides that kill pretty much everything, followed by selective herbicides, which only have effects on one broad group of plants, such as monocot versus dicot, or perennial versus annual. Since this is an agricultural channel, mostly anyways, it will only cover herbicides used in agriculture or plant production. We will start with paraquat and diquat. These are both bipyrimidium-based herbicides. Diquat was first created in the late 1950s, and the former was created in 1962. Paraquat persists a lot longer in the soil than diquat, and both are very toxic to humans and really all mammals. The mode of action of paraquat is basically chemical lands on the leaf and reacts to sunlight, creating free radicals. The free radicals burn through the cells and eventually kill the plant. As of right now, paraquat is not used very much in the Western world, but it's still used in developing countries. Now on to glyphosate. It was originally marketed as a cleaning agent, but it was soon found that it was really useful for killing weeds. Its mode of action is that it inhibits the ESPS enzyme within plants, both preventing the creation of different amino acids that the plant needs to survive, and creating free radicals that damage a plant even further. These two processes eventually kill the plant. Now on to glufosinate. Glufosinate was created in the 1970s by combining left-handed and right-handed versions of phosphine-oricin. This chemical prevents the synthesis of an important plant chemical called glutamine, which kills the plant. Like glyphosate, it is non-selective and kills all plants equally, so genetic engineering was used to make crops immune to said pesticide in order to make it suitable for agriculture. In the case of glyphosate, there's many different ESPS enzymes, and such enzymes are found in fungi, bacteria, and plants, but not in animals. Hence why we are not affected by glyphosate in the same way plants are. One bacterial species, however, had an ESPS enzyme that was not affected by glyphosate. The gene that codes for this version of the ESPS enzyme was then used to replace the genes within corn and soybean genes for ESPS, thus granting immunity to this herbicide. In the case of glufosinate, a species called Streptomyces had developed interesting genes called BAR or PAT genes that can detoxify phosphophenorycin. Those genes were encoded into the plant through genetic engineering, granting immunity to glufosinate. Now on to selective herbicides. In the 1940s, researchers discovered the plant hormone IAA. which controls plant growth. It was also discovered that high concentrations can stop plant growth or create irregular plant growth that can kill the plant. However, it only works on broadleaf plants, making it suitable for use in fields that grow grass-based crops, such as corn or wheat. So researchers began looking for more powerful analogs of IAA. The first discovered one was MCPA, which wipes out all broadleaf plants within a field, leaving only grasses such as barley and oats, the actual crops. A derivative called MCPB was safer in that it did not work on legume plants. A later version called 2,4-D, made in 1945, was also created. This was called 2,4-D, and it works in much the same way, but at a more potent level. Due to a volatility issue, a new version called 2,4-D-C was made to be less volatile. This, in combination with semi-dwarf wheat varieties and fungicides, increased wheat yields by a considerable margin. A later derivative of 2,4-D called 2,4,5-T was developed to deal with woody weeds, and was first used in Agent Orange in combination with 2,4-D to destroy forest cover during the Vietnam War. However, it was found that there is a chemical contaminant made as a result of the manufacturing process for 2,4,5-T, and this contaminant caused cancer. This chemical is called TCDD. Now on to atrazine. Atrazine inhibits photosynthesis at photosystem 2, one of the two systems used to produce photosynthesis. Corn, however, can detoxify this herbicide, making it useful for controlling weeds within a cornfield. Next is dicamba. Dicamba is first found during the development of 2,4-D as an offshoot of the research. It was, however, far too volatile to be used. With the advent of brand new herbicide-resistant weeds, such as Canada fleabane, new herbicides to kill these plants were needed, resulting in dicamba being released. Due to the volatility, it needs to be used a lot more carefully since it can damage non-resistant crops downwind from the spray very easily. And various technologies to combine them in different formulations, such as the vapor grip technology, were made specifically to make a less volatile version of dicamba. Now on to our final herbicide, the sulfonylureas. This group of herbicides are acetolactase or ALS inhibitors. By inhibiting the ALS chemical, the plant is unable to make the amino acids valine, isoleucine, and leucine. These three are necessary amino acids to make DNA. Without these amino acids, no more DNA can be made, and the plant dies. This specific chemical that works on ALS only kills broadleaf weeds that are annual in nature, although there's other ALS inhibitors that work on grass weeds as well. Well, that about covers everything. Thank you for watching. Hello and welcome to the third episode of Pesticide History. In this episode we'll cover fungicides. The earliest known fungicide was called the Bordeaux mixture. It was initially created as a means of preventing fruit theft, but was found to also control fungi. It was a mixture of copper sulfate, hydrated lime, and a lot of water. In the first era of fungicides, it was entirely consistent of inorganic metals being mixed with water and other compounds as a means of controlling fungus, with later versions being coupled with organic compounds to improve the sticking potential. Later discovered that you can culture fungi in petri dishes and use different chemicals on the fungi to see if they can kill the fungi. This led to the creation of new fungicides, although it also required safety testing. This happened in the 60s. Most common forms of fungicide were developed as the organic protectant fungicides, which were developed in the second era of fungicides, and the systemic fungicides, which were developed in the third and current era of fungicides. We will cover both in this video. You'll start with the organic protectant fungicides. It was first discovered in 1934 that diethyl carbamates had antifungal properties, although no commercial development occurred until after the Second World War. The first fungicide used commercially was Furbam, a ferric salt form of diphocarbomatic acid, although it became unpopular due to the fact that it left a black deposit on leaves. A later derivative, called ferrum, was later discovered and it lacked the black residue but still had equal to or greater resilience than the previous fungicide. It was used extensively on the fungus known as Pythium, as well as other pathogens of fungal origin. Another strain of this same chemical combination called axial-diamines were made and were often coupled with zinc to create Zinleb or manganese to make Manleb, or a combination of the two called Mancozeb. These strains of fungicide had low negative effects on the plants themselves and could protect against a wide range of fungal ailments, with the exception of powdery mildews. Mancozeb was first created in 1961 and remains in the EU and is still used to this day to deal with different diseases in potatoes, ornamentals, and tomatoes. Still, yet another class of fungicides called the quinones were also created, although they were unstable in light. These were often mixed into soil to deal with root infections. The most popular form is called Captan. The first one was introduced in 1952. It is used extensively to treat apple scab disease and a range of vegetables and ornamentals, and it deals with a wide range of different fungal species. Another group introduced in the 1940s were called the nitrobenzenes. Due to their volatility, they're not used in the field regularly, but they are used extensively in preserving fruit post-harvest. In 1959, the chemical Cyprex was developed and had significant abilities to eradicate fungi, and is very mobile on the surface of plant tissues. Introduced in the 1964 era, and is used to this day mixed with other fungicides. On to systemic fungicides. During the 20th century, the holy grail of fungicide research was to discover chemicals that could move within the plant, to act as a means of killing the fungus once an infection happens, and to act as a protectant, much the same way organophosphates and neonics work. The first one discovered was grisofluin. This was found in the 1940s. It moved throughout the plant within the plant tissue once applied, and gave control to some diseases, but was too unstable for general use. A later one discovered called acidione was found to be really good at controlling white pine blister rust. And in 1959, 4 million trees were sprayed with this fungicide, and it controlled the disease, but also caused a lot of damage to the trees as well. In the 1960s, various chemical companies such as DuPont, ICI, and BASF, and Uniroyal began making their own systemic fungicides through different research processes. DuPont discovered benamyl in 1968, around the same time that ICI discovered dimethyl in 1968. BASF also discovered dodomorph in 1967, and Uniroyal discovered carboxin in 1966. Benamyl was the first of the fungicides to be used, and it was used extensively against a range of diseases in cereals, orchard fruit, vegetables, and vines. Unfortunately, it was also toxic to other microorganisms and invertebrates, especially earthworms, and was in fact used to kill worms prior to being used as a fungicide. Fortunately, due to this fungicide only attacking one site of action, meaning it only attacked one chemical pathway within the fungus, the fungal strains quickly adapted through the process of evolution to develop resistant strains of fungus to this fungicide. One such example being Cercospora, or the leaf spot fungus, which is found on both sugar beet and wheat. Dimethyl, used extensively on powdery mildews in fruit crops and ornamentals, but like benamyl before it, only attacked one chemical pathway, and so the mildew developed resistance to it very quickly. Carboxin came afterwards, and was used to treat rhizoctonia, and is still being used to this day because the smut fungus cannot withstand this fungicide very well, for unknown reasons. Unfortunately, none of them worked against the oomycete fungi which are not actually fungi. Turns out that oomycete fungi are actually a different kingdom of life altogether. We'll cover how they dealt with that next. First fungicide to deal with oomycetes, such as Pythium and Phytophthora, and downy mildews, the phenylamines consisted of metaaxyl, furaxyl, and betaxyl. They worked against all oomycete species, but are ineffective against regular fungi. Also worked really well against potato blight, which is also oomycete in nature. Later on in the mid-70s, a triazole group was also developed to deal with oomycetes. Also worked on different types of funguses as well, such as smut and one leaf blight species. Two forms were later created to make. Later forms were also used to control other fungi species such as anthracnose and other cereal pathogens like eye spot. As more formulations were added, more fungi were added to the hit list, including different types of rust. This continued to be developed up, and research continues to this day. In the 90s, another group of fungicides called strobilurins were also created. First engineered from various fungal poisons found ironically in mushroom caps. They're used extensively on different diseases such as scab, downy mildew, clubroot, white mold, and alternaria. Now on to the mode of action for all these fungicides. The carboxin chemical fungicides disrupt a chemical pathway within the electron transport chain of the mitochondria called succcinate dehydrogenase of complex 2, effectively disabling their mitochondria, preventing ATP from being formed, killing the fungus. The dodomorph stopped the synthesis of rhizobium RNA, and the majority of protectant fungicides have multiple sites targeted, meaning that these older chemistries disable multiple chemical pathways within a fungus. The most systemic fungicides use similar pathways to disable the fungus, leading to cross-resistance, requiring a combination of different fungicides to be used to treat different diseases, prevent resistance. Well, that about covers everything. Thank you for watching. Hello and welcome to the series finale for Pesticide History. In this episode, we'll cover biopesticides, their mechanisms, and history. Let's begin. The first one we'll cover is Bt. After World War II, a bacteria called Bacillus thuringiensis, or Bt, was discovered. It was found in the diseased larvae in the Mediterranean flower moth. It had limited viability, however, as effective pesticides simply because the air synthetic pesticides worked a lot better, although it found use in genetic modification. Another biological control was the fungus called metarrhizum. The species M. arachium was found to be effective in controlling locusts in Africa, and could be used in a low-tech spray equipment method by mixing it with vegetable oil and using an ULV spray technology. This species of fungus was also used to deal with grasshoppers in Australia, and another species discovered was used to treat vine weevils on strawberries and other soft fruit crops. Bt likewise was developed further, with several subspecies being found that could be used to control different types of caterpillar pests. In later days, the genes then code for the proteins that are used by the fungus to kill different types of caterpillars were incorporated into plants via genetic engineering, allowing the plant to directly kill their pests. Another biocontrol is Bavaria bassiana. The biopesticide is used for greenhouses to control whitefly. It's once again a fungus, and its fungal species feeds on most insects. There have also been attempts to culture viruses to deal with insect pests, such as bacilloviruses. These biopesticides, however, are very expensive due to the need of living insects to culture the virus in, although a few species of this virus are available for use to deal with codling moth, gypsy moth, leafworm, and cassava hornworm. Now onto biofungicides. There are a litany of microbe species being tested out to deal with various species of fungus that are soil-borne, some of which have been released to public use. The first one we'll cover is Conanrifrum minetans. This is the only biocontrol agent registered in Germany as a biopesticide. This is a mycoparasite that consumes white mold. It was initially tested in England and was found to consume the fungus on a number of different substrates such as barley and wheat. And this mycoparasite infects 80% of the white mold it comes in contact with. The next one on our list would be Glaucladum virens. This species was found to be able to control Pythium and Rhizoctonia pathogens in soilless mixes within greenhouses. It was cultured by the USDA in the late 1980s. It was first discovered in a soil sample in Maryland. This fungus produces as a waste product the toxic compounds glyovirin and glyotoxin, both of which are very toxic to Pythium and Rhizoctonia. In essence, the fungus's poop is poisonous to other fungi. The next species on our hit list is Trichoderma harazium, strain T22. The base species parasitizes different fungal species, although those that dwell near roots do not consume as many fungi as the ones found in wild soil. Strain T22 was made by fusing the cells of T95, a strain of the species that's found in the root system of different plants, and the strain called T12, found in just regular soil. The result of this fusion allowed for a strain of this parasite that can survive and thrive and mass produce in the root system of plants while rapidly consuming any other fungal species around. It's commonly used as the seed treatment known as root shield, which provides control equal to fungicides when applied as a seed treatment, as a preventative. It is commonly used in various ornamentals like poinsettia, geranium, and impatiens. It also competes with other colonizing fungi and bacteria, and also causes an induced resistance to different fungi within its host plant. Now on to Streptomyces geoverdis, known by the trade name Mysostop. This is a parasite that was found in sphagnum peat moss and became researched and eventually found to be useful for controlling fusarium wilt in basil and carnations and Douglas fir. Next on our hit list will be Glaucladium kinatulutum. This species was found in Finnish field soil and was used as the active ingredient in Primostop. It seems to be effective against different types of root pathogens such as damping off, seed rot, and root rot. As of right now, it's used mostly in ornamental bedding plants, although effectiveness varies. The last one we will cover is non-pathogenic Fusarium oxysporum, strain FO47. Initially found in a soil that had no Fusarium infections in France, it was discovered that that landmass had a strain of Fusarium that rapidly inoculated the plant but did not cause any damage. Basically, it's a fast-growing, asymptomatic variant of Fusarium. This was then cultured out and used to treat different types of diseases as a preventative within greenhouses, as well as outdoor tomato and ornamental growth. The main mechanisms of action include the induction of host defenses and direct competition of other strains of Fusarium. This strain was found in that soil, as I mentioned before. This landmass was studied for over 25 years by the organization INRA, and this Fusarium species is a culmination of that 25 years of work. Well, that about covers everything. Thank you for watching.